Good to have you here. I want to welcome you to Bodega Marine Lab and also to the inaugural lecture of the Dr. Donald L. Michaels Distinguished Lecture Series. It's a big day for us, so it's cool to see you all here. Thank you for joining us. I'm John Lagier. I'm the director of Bodega Marine Lab and the professor of oceanography. Not about me, but I get the pleasure to welcome you and to invite you to Davis by the Sea. We kind of think of ourselves as the center of marine science in Davis for people on campus, I argue. But this is sort of the beating heart of where a lot of the, are you hearing me okay? Should hold up here. This is where a lot of the experimental research is happening, both in wet labs and aquaria, out in the field, collecting data, doing experiments, tracking changing ocean conditions, and it's an exciting place for all us to be. We combine this with classes, undergraduate classes. There are many students here right now during summertime, and resident graduate students as well. But we also combine it with an engagement with our community here in West Sonoma County. So this is a wonderful opportunity to do exactly that. This is the inaugural event is made possible by the generous donation from the Michaels family, who are here today. So welcome Vicky, and Christopher, I don't believe is here, but he's on Zoom, I believe. The series is named in recognition of Don Michaels and his work in marine biology, which has been anchored here in Bodega Marine Lab for decades. Good example of the value of having a place-based research. Their aim is to bring an accomplished scientist to Bodega Bay each year, and through doing so to honor and elevate that person's work, as well as to invigorate Bodega Marine Lab and to share the excitement and intrigues of marine biology with our community here in West Sonoma. We also hope to develop lifelong partnerships and collaborations as exemplified what we've had with Dr. Michaels all through Don's career. He started here in the 1970s as a grad student and has visited annually since then. Bodega Marine Lab is very grateful that this Michaels Distinguished Lecture Series has been funded through a continuing gift from Vicky and Christopher Michaels. They invite you to join them as partners by making a gift of any size to this program. There's an opportunity to do so on the invite, and there are QR codes around the room and the building. Working together, we can secure this important lecture series for the future, and for marine and coastal scientists and for the broader community. So I've got papers in the wrong place. So I'm going to talk about Don for a little while. He earned his PhD in 1979 from Berkeley, and during that time he was a graduate student resident here. He went on to a postdoctoral position at Tennessee Oak Ridge National Lab, and in 1985, he took a position at Colorado State University where he established the Crab Lab. In 1998, he came back and spent a sabbatical year working here with Ernie Chang and began a nearly 20-year collaboration aspects of crustacean physiology. Don has had a very successful research career with major research grants from Sea Grant, NIH, NSF, and other sources. Internationally respected as an expert in the field of crustacean molecular and cellular molting physiology. He's been many things: a Fulbright Scholar, a AAAS Fellow, and has published many scientific papers, many of them reporting results from research conducted here. So today, in addition to welcome you all to BML, we're honoring Dr. Melissa Pesmany. Dr. Pesmany is hiding behind a banner on her head. Interesting. Called Rick Grossberg, which is kind of apt. He raised his hand. Probably he wants me to hold the microphone higher, right? Not getting picked up by Zoom. So welcome, Melissa, Dr. Pesmany. Dr. Pesmany is a creative and dynamic evolutionary biologist who applies cutting-edge genomic, genetic, and computational tools to address some of the most important and timely challenges in biology today. In particular, much of her research asks, to what extent can marine species adapt to climate change and disease? Seems like an important question. Dr. Pesmany received a PhD from Stanford University in 2010 A dissertation addressed whether purple sea urchin populations are locally adapted to environmental conditions along the west coast of North America. Dissertation was very influential because it's among the first to apply these new genomic methods to reveal that there's a lot more selection and population differentiation going on in coastal species than we had appreciated. After receiving her PhD, Dr. Pesmany stayed on at Stanford as a postdoc, researching whether sea urchins have the capacity to adapt to ocean acidification. She cultured sea urchin larvae here at Bodega Marine Lab under different pH levels and used next-generation sequencing to uncover striking shifts across the genome in response to selection imposed by acidified conditions. Dr. Pesmany further expanded her range of interests and approaches during a second postdoc at Indiana University, where she investigated the evolution of novel trays in horned beetles, well-known marine species, which extended her skills in genomics and genetics into the realm of developmental biology. Dr. Pesmany was hired as a faculty member, University of Vermont in 2014, where she established a dynamic research program focused on climate change adaptation in coastal marine ecosystems. She received a prestigious NSF Career Award, and she's been using innovative selection experiments and genomics to understand the scope of adaptation within marine populations. Beyond her stellar research achievements, she has a deep commitment to teaching and mentoring. For the past 6 years, she has served as director of an innovative graduate training program at Vermont called Quest. She's also worked actively to advance diversity, equity, inclusion through mentoring, workshops, and a variety of efforts at University of Vermont and beyond. While Melissa is considered a rising star, her visit here is more than an honor. It's a profound contribution to the Bodega Marine Lab and a work in progress. She has brought her insight, her skills, experience, her mentorship, and dare I say, her heart to our community. And we have many different activities going on through the week, which has been wonderful and will continue to be wonderful. Interacting with many of us who are resident here and also interacting with graduate students and undergraduate students and other visitors to the lab. I could go on, but you're here to hear Melissa. So without further ado, Melissa, I congratulate you on the honor and I invite you to share with us your novel insights to the ocean and marine biology.
Thank you, John. All right, lovely. Can everyone hear me all right? Okay, all right. Oh, we didn't start the video over here, but that's okay. We're getting this. All right. Thank you so much, John, for that lovely introduction. And thank you, Vicki and Dr. Michaels. Where are you? Okay. Thank you for this idea, for this inspiration, for having this forum, to have a community talk. It's just a wonderful idea. And I guess most importantly, thank you all for choosing to come here tonight, to be here as the community members. If you weren't here, there would be no community talk. So thank you. So we live on this incredible blue marble of a planet with breathtaking biodiversity. In every nook and cranny, from mountaintops to deep ocean trenches, we have incredible organisms living in and around us. In the sea around us, actually, to borrow a phrase from Rachel Carson. But this, and some of the examples that you'll hear about today are purple urchins living in cracks and crevices or mowing down kelp forests, whether we want them to or not. Sea stars making way for incredible biodiversity in the rocky intertidal. And these 1 millimeter long copepods that are the base of the marine food web. But this climate is changing at a geologically unprecedented rate, which is a problem for many marine organisms. So not only are global temperatures rising, but also it's resulting in changing in weather patterns. So increased floods, droughts, fires, things that we unfortunately know about all too well anymore. But also the Earth's oceans kindly absorb about 1/3 of that CO2, which is wonderful for us on the land, but problematic for a lot of the marine organisms that need to be able to grow and maintain skeletons. We're also putting a number of things in the oceans that were never there before, like plastics, like antibiotics from our wastewaters, like runoff from agricultural fields. So there's lots happening. But as an evolutionary biologist and as a lover of life and as a lover of biology, I want to know how these organisms have adapted to this incredible environment and how in turn they might be able to use these mechanisms to adapt to future global change conditions. But before I go any further, I want to share with you this photo that my mom just shared with me recently. I asked her to send me some photos from when I was a kid because I was doing a presentation at one of my kids' schools. And you can see the spark. You can see that I'm turning that poor creature upside down. So I had the pleasure of growing up very close to nature, growing up in Okinawa, Japan, with torturing geckos in my backyard and damselfish in my front yard. And then I moved to California shortly before this picture was taken, and I was floored by the graffiti and the gumboot chitons. So it's a very incredible and inspiring place. But this spark that you can see here, the fact that I'm turning this over, I've always wanted to know how things work. But I know that each of you, as community members that chose to come here tonight, are people that have that spark too. So if you feel comfortable, I'd like you to close your eyes for a second and think of that organism, that place, or that person that inspired in you your spark or your curiosity. Helped you cultivate that curiosity. So think of this situation, the organism, the place, the person, and notice the quality of the air around you in that environment. Notice any smells, notice any colors, notice any textures, any sounds. Just relish in that spark. Okay, bat your eyes open if you close them, and hang on to that curiosity, because I'm about to share with you lots on mechanisms, and you'll need some curiosity for that. I'm going to start with an analogy of DNA as a musical score to help us all get on the same page of these mechanisms. So if DNA is a score, if you can think of the notes on a page as a string of nucleotides, that would be the musical score. If that's the musical score, then the genes are all the different musicians playing each of their parts. And then naturally, the incredible art of playing that music would be gene expression. Then the whole genome would be that whole composition, so that all those notes strung together. And continuing the analogy, the whole concert hall would be the organism, and the environment would be the lights and the acoustics and the temperature in that room, in that space. But what's the variation? Those will be the tools that allow an organism to survive. So the variation would be the differences in how the orchestra plays the music from night to night, or from orchestra to orchestra. Those would be the DNA mutations that happen. And epigenetic variation would be the dynamics that are noted above the musical notes that affect the dynamics, so how loud or how soft or how fast or how slow the music is played would be the epigenetic variation. And there's one more piece of variation that I'll talk about today, which is the microbiome. So the microbiome in this analogy would be the audience. So I'm sorry, you are all the microbiome. You are the microbes that live in and on the host. So in the concert hall analogy, it would be the regulars, the patrons would be those that are the core microbiome, and those that come, you know, same day ticket buyers or passersby that got in, those would be the transient or the opportunistic microbes. So this is the variation that natural selection has to work with. So the genetic variation, the epigenetic variation, the gene expression, the microbiome, these are the things that can affect the health and fitness of an organism. And these are the things, so, okay, one more piece to the analogy, and I'll stop, is that would be the sellout nights, the number of sellout nights and the standing ovations. That would be high fitness for in this analogy. And just to show the DNA mutations, I'll indicate, like with this blue mark here, and the epigenetic mutations on the outside of the DNA here. So this is our classic double helix of a DNA molecule. Let's introduce our first character, the purple sea urchin. So it's kind of a nemesis around here with the loss of their predators and the changing of global conditions. They can do an amazing job mowing down a kelp forest. But for the study of evolutionary biology, they are a wonderful model organism, because they have this broad latitudinal distribution from the cold waters of Alaska all the way down to the warmer waters of Baja California, Mexico, which is an environment that they could perhaps adapt to. So there's environmental variation. They also have dramatically large population sizes, as you can get a sense for in this photo, which allows them to harbor genetic variation that could be substrate for adapting to future environmental changes. And they also have these dispersing larvae, as you can see in this image here, that grow skeletons, that need to be able to grow skeletons in order to feed, in order to be able to swim, because they're ciliated and help them feed and swim. These dispersing larvae would be a nightmare, essentially, for local adaptation, because they mix all the genes up and down the coast. But this is unless you can look across the whole genome. Then they're the perfect organism, because genes that have allowed them to adapt to local environments will then stand out like a sore thumb against this background of gene flow. So that's the purple urchin. And what one of my graduate students, Chenga Patek, wanted to know was how they can adapt to environmental variability. Uh-oh, it's not showing up here. Okay. So she sampled 20 urchins from each of 7 populations from Fogarty Creek, Oregon, down to San Diego, California, and sequenced their whole genomes. And she wanted to find genetic variants that would be associated with pH variability. So not mean pH, but the degree of pH, or the time spent at really low, or the frequency of time spent at low pH conditions. So she found all the genetic variants that were associated with that. And it turns out that many of them are in the regulatory regions of genes. So here's our double helix, here's the genic region, and just above that is what we call regulatory region. So it controls whether or not, when and where a gene is turned on. So she found, as you might expect, when you're needing to adapt to variable environments, that a lot of that variation was in regulatory regions. And you can kind of think of it as a light switch with a dimmer that can be turned on or off or dimmed to any degree. She also found, there's a delay here. It's on my slide, but not there yet. Okay, there we go. So we also found that if you look at all the genes that are related to growing a skeleton in a sea urchin, so these are called biomineralization genes, that you get this strong clustering by how much time each population spends at very variable pH conditions or at low variability pH conditions. So this clustering, based on how much time they spend at low pH conditions, suggests that these genes are very important for being able to adapt to variable pH conditions. And speaking of that sore thumb, so this would be the sore thumb, because when we look across the whole genome, you can see that there's no clustering based on how much time is spent at low pH conditions. So these are what we call phylogenetic trees based on, say, a million genetic variants identified across the whole genome. But what about ability to be able to use this genetic variation to survive changing climate conditions? So that's what Eric and I wanted to address many years ago. It was about, I think, 12 years ago now, that we thought of this way to test for to identify genetic variation that might be able to help organisms survive in low pH conditions. So we spawned urchins, as you can see here, and we reared up millions of sea urchin babies and looked at them under the microscope to see how they're growing, how they're developing, but also what genetic changes might be occurring. And that technique led to lots of great studies that have continued in my lab. This is a funny delay. Okay. So the technique is essentially this. You capture a bunch of genetic variation based on sampling urchins in the wild, you spawn them, bring them into the lab, make millions of urchin babies, and rear them up in different conditions, and then you can see who survived. So we continue to do this in my lab in Vermont. Oh, why is it taking so long? It's the internet. Oh, okay. So we continued to do these experiments in Vermont where we can rear the sea urchin embryos in various pH conditions. So these are some of the people carrying out the experiments, and what we can see here is rearing them in ambient pH conditions. So this is open ocean pH 8.2, lower pH, slightly more acidic, and much more acidic pH 7.5. And what we found is that these are our sea urchin babies, and this is the degree of mortality. We also measure growth. You can see higher mortality, but more importantly, we can measure the genetic variants that allow them to survive. And we find that it's the rare variants that allow organisms, or the sea urchin babies, to survive these really extreme pH conditions, whereas it's the common variants that are important for both. And what this translates to is that rare variants are maintained in populations that have lots of individuals. So large population sizes are important for surviving extreme low pH events. But common variants are important for surviving the both pH events, and those are maintained in populations by having environmental variation. So you need that degree of environmental variation to have common genetic variation. So both large populations and common genetic variation are critical for purple urchins to survive, but this is also something that we can translate and extend to other marine organisms. So whatever we can do to maintain population sizes such that organisms have lots of individuals, can harbor lots of genetic variation, but also that they experience the rate or the degree of environmental variation that they typically do will help organisms. So another incredible way to study adaptive capacity is to look across generations. So the coastal copepod, Acarcia tonsa, is a wonderful organism for being able to look across generations. They are also at the base of the marine food web. So they are the organisms that eat the phytoplankton, and then they themselves are eaten by baby fish. So they're at this critical point. They also result in the carbon sinking of, so all of that phytoplankton that they eat, they then poop out, which results in the sinking and sequestration of carbon, which is quite helpful to our biogeochemical cycles, even though we don't even necessarily think of it or appreciate it as we walk around on Earth or on the land. The other thing that's fabulous about them is that it takes just 20 days to go from egg to reproductive adult. So in some of the most beautiful local adaptation data that I've ever seen was carried out by a recent graduate from my lab, Lauren Ashlock, who some of you may know, who is now a postdoc in the White Abalone Conservation Group. So she did a very clever experiment where she brought organisms, she went out and collected in the field. This picture is from Maine, I believe, right, Lauren? And she collected organisms from Maine, New York, and Florida, and brought them in the lab, reared them for a couple generations to erase the environmental history that they had, so she could look at what are the genetically controlled differences in their thermal tolerance. So what you're looking at here is their upper lethal temperature, which is a proxy for how well they can survive in warm temperatures. And you're looking across 3 different life stages. So the babies, the teenagers, and the adults. And you can see, if you look across the populations, there's much higher thermal tolerance in the organisms that are from Florida, for example, and then lower for New York and lower further yet for Maine. So this gave us an idea that there are signatures of local adaptation in these copepods. We wanted to know, can they use that genetic variation to adapt to future global change conditions? And we're about to get at the mechanisms of this here with measuring the gene expression with 2 colleagues of Lauren's, also in my lab, so graduate student Allison Hall and research technician Manali Rika Colt. We're here doing apple picking, our annual apple picking that we get to do in Vermont. And we'll soon know the mechanisms underlying that. But we wanted to use this pattern of local adaptation, or knowing that there's genetic variation, and experimentally evolve them. So we designed an experiment where we would rear them in ambient warming, so from 18 to 22 degrees, then also acidification, so from 400 to 2,000 microatmospheres of CO2, and then the combined warming and acidification. So ocean warming and acidification will abbreviate as OWA. We had 4 replicates from each of these. In each of these replicates, we had about 5,000 copepods acting as a population. So we experimentally evolved them for some 25 generations and measured fitness-related traits. So how many eggs do they produce? How long do they survive? And then we also sequenced the genomes of these pools of copepods. And what we found was, after just 1 generation in these environments, the organisms in ocean warming and acidification had a dramatic decline in their population fitness due to decreased egg production, hatching success, survival, etc. But the good news was that after just 3 generations, they recovered to the same levels as the organisms that were in ambient conditions. And this kind of bounced along for a while until about 25 generations, and there was a bit of a decline. We saw, and we wanted to know, that those were the data on their phenotype. We wanted to know what's happening genetically. Was this all genetically controlled, or is it just a degree of flexibility that they have? So we did an analysis where we, looking at all the genetic variation, we do an analysis called principal components analysis. And you can think of a massive data matrix here, shown in this table, and we've got the samples as all of the columns. So here's the 4 replicates from ambient, 2 of the replicates from ocean warming and acidification, and the table continues for all of these. And then for all 500,000 genetic variants that we found, we can use clever math to be able to see how similar or different are each of these samples and plot them in this space called principal component space. And this first axis, 1 down here, will explain most of the variation. And then in this case explains about 15 or 16% of the variation. And the second one explains about 11%. So when samples are more similar to each other, they will cluster together. When they're more different, they'll be further apart. So this is what we found. That there was strong clustering by each of the treatment groups, suggesting a strong, consistent genetic change in each of these treatment groups across the 4 replicates of each. and the strongest signature being between the founding population and warming, or ocean warming and acidification, and then warming, and then also a signature of lab adaptation happening, and very minimal differences between ambient and acidification alone. So there's a strong signature of adaptive genetic divergence happening. And most of these are happening, most of these genetic changes, if we count the number of genetic variants that are unique, most of them are happening in the ocean warming and acidification, followed by warming alone, and then ambient, the lab adaptation, then acidification. And if you look at the shared variance between ocean warming and acidification and warming, that also has a lot, and then declines. And what are these genes? This is the how. This is me turning over the gumboot chitin. This is for adapting to the ocean warming and acidification. It's ion homeostasis and regulation of cell growth and energy processes. So being able to control your ion balance and control your energy processes. And for the warming alone, it's tissue homeostasis, so maintaining the stability of your tissues, and regulation of gene expression, and regulation of development. So we show that they go through this ring of fire. We show that going through this ring of fire, they actually don't lose any genetic diversity as they're adapting. But we wanted to test if there in fact are any costs to this. So we did what's called a reciprocal transplant, or swapped them. So our ambient animal lines here in blue, and our ocean warming and acidification in red. And after 20 generations, we took animals from ambient and put them in the ocean warming and acidification as a control. And we also returned animals from ambient, or from ocean warming and acidification, and put them in ambient. And these symbols you'll see here for the blue circles are ambient animals, the red triangles are the long-term ocean warming and acidification animals, and then the swapping environment. The color indicates the environment that they're in, the shape indicates the selection line that they were a part of. So blue triangle was moved into the ambient environment, red circle was moved into the ocean warming and acidification environment. So we do a really fun analysis that's similar to the one I just described to you with the principal component, but we can polarize based on these samples. So we expect the greatest differences between the ambient and the ocean warming and acidification. We want to see where do the transplants fall? What happens to them? And in this example, you can see that just after one generation, the ambient animals adapt to the ocean warming and acidification environment to quite a degree. We're looking at gene expression patterns here now instead of the genetic variants. So their expression of all the genes matches this ocean warming, or gets towards matching that environment. For the ocean warming and acidification animals, there's barely a change once they're moved to ambient, at least at first. So these are the same data for generation 1 here. So you see this major change. Generation 2, now the ocean warming and acidification animals are starting to make it across towards this blue ambient adapted space. And then by the 3rd generation, they're quite far over, as are the transplanted ambient animals. So this shows that there can be this rapid readaptation to the alternative environment. But when we look at how they did it, it turns out that the animals from the ambient environment handled the change to ocean warming and acidification easily. Just without any genetic change, they were able to just change their gene expression. But the other animals moving from the ocean warming and acidification to the ambient environment had to re-genetically adapt to that environment. And when they did so, they lost genetic diversity. So this is these ocean warming and acidification animals moved into ambient conditions. This is generation 1, 2, 3. You can see by the 3rd generation, they lost a lot of genetic diversity, and they lost diversity in the very genes that they used to adapt to that environment, that ocean warming and acidification environment in the first place, which is problematic. The other thing we looked at was whether or not there could be epigenetics would play an important role in this. And we found that while there's not as strong of clustering as we saw with the genetic variation, there is indeed a signature of a treatment effect and adaptive or adaptive epigenetic divergence in response to these 4 different treatment environments. But what was really interesting is if we look at where they fall in the genome, it turns out that all of the genetic variation was in certain regions and the epigenetic variation was in totally different regions. So not overlap, suggesting that there's important, distinct mechanisms for each of these processes, genetic and epigenetic variation. One more story with the copepods. This was a student who started as an undergraduate in my lab and stayed on as a master's and came in really interested about microplastics and plastics. So everything that ends up in the ocean that's plastic degrades and ends up being microplastics, little bits of plastics. That organisms like copepods that need to feed on microalgae can mistake them as food because they end up just the same size as food. So what she did was reared them in with algae and with microplastics, so they had both. They ate a lot of the microplastics accidentally. So here you're looking at the bottom of a copepod. Here are eggs. Here is a fecal pellet about to come out. And she found that growing them with the microplastics, they had overall reduced growth, reduced survival, reduced egg diameter. And then over time, we put that all together and modeled that over time, their population growth rate would just dwindle, which would be quite problematic for our ecosystems. And then she also noticed, she spent a lot of time looking at this copepod poop and found that it was very fragile. It would break, it was much more brittle and would fall apart. So not only did they have smaller volume and less total matter, but it was more fragmented and therefore would sink at a slower rate, which would, as we did some modeling for this, result in reduced carbon settling, which again would be problematic for our Earth's oceans. So I've shown you that copepods can indeed adapt to these global change conditions using genetic and epigenetic mechanisms, but there's limitations to this adaptive capacity. There could be a point which they get to the end of the line or the end of the pier, as it were, and might not have enough genetic material to continue to adapt as conditions change or as a novel stressor presents itself. A novel stressor such as, for example, a new disease pressure, which leads me to sea star wasting disease. So many of you know and are familiar with sea star wasting disease, but it's known as or called the largest marine epidemic in the history of our recordings. Not only because of how gruesome it is, but because of the large geographic extent from Alaska down to Baja California, Mexico, the number of species, some 20 species affected, and the near 100% mortality of some species at some sites. So what we wanted to know was, why now? These symptoms have presented themselves at some points in the past. Why had it now become to such a geographic extent, affecting so many species and causing such mortality? So one thing that hadn't really been considered was the role of the microbiome. And we know that microbes play an important role in health and disease of many organisms, including humans, and including corals. So in coral black band disease, or in humans with pneumonia, or irritable bowel disease, or even allergies. There can be symptoms that are caused by many microbes and caused by an imbalance in the microbial community. So we wanted to see if that was happening. So we did some lab studies, some lab time course studies where we brought 37 animals into the lab that were Pisaster ochraceus, and we were going to do a quarantine and experimental infection, but they started showing symptoms right away, so the experiment started right away. And every 3 days, we sampled them for a period of 2 weeks. And what we found here is this progression of symptoms. So from 0 to, we characterize them as 0 being totally healthy, 1 showing 1 lesion, 2 showing 2 lesions, 3 showing 3 lesions, 4 an arm pulling off at least, and 5 death. And looking through this 2-week period, you can see all of the animals arrived overnight healthy and asymptomatic, but then progressively through the 2-week period, most of them succumbed to wasting disease, with only 8 surviving. And looking at the microbial community composition, this is a plot where we have the most abundant microbes, including this category of uncharacterized microbes, unknown. And we're looking at the proportion in these samples from sea stars that either were 0, 1, 2, 3, 4, or 5 conditions or symptoms. And I'll point out just a couple things. There was a dramatic decrease in these putatively beneficial unknown microbes over the course of symptom progression. And there was also an increase in putatively pathogenic or opportunistic microbes in these late stages of disease. We also importantly showed that for the animals that stayed healthy, they were very resilient. Their microbial communities were very resilient to the lab conditions. So it's not just an artifact of being in the lab. We also wanted to know what's happening with the host. So we measured their gene expression. We found that in the animals that were healthy, they had much higher expression of genes related to maintaining their collagen, maintaining their tissue integrity. So these are these pro-collagen genes. They also had really high expression of immune genes. We thought that maybe we were going to see the opposite, that animals experiencing wasting would have higher expression of immune genes, but no. We found that the healthy animals were eliciting an immune response, or responding immunely. In the wasting animals, we found higher expression of RNA processing genes, a number of which have been implicated in neurodegenerative diseases in humans and other organisms, suggesting, interestingly, that perhaps the loss of control of their collagen system, which is a neurologically controlled system, could be controlled by these RNA processing genes. So just a symbol of what's happening to them. We also saw much higher expression of this hypoxia regulator, so a master gene regulator called hypoxia-inducible factor 1. And I'll show you just what those genes look like briefly here. Here's this hypoxia-inducible factor 1 alpha, where all of these orange dots are samples from a healthy sea star. In purple are showing one of these symptoms. You can see higher expression, particularly given that this axis here is log scale, so much higher expression in the animals experiencing wasting. So there's evidence that low oxygen conditions are perhaps stressing out these sea stars. Similarly, this is an enzyme that breaks down collagen. So it's not a pro-collagen, but it breaks down, and again, it's higher in the wasting sea stars. And unfortunately, we looked at the genetic variants and found no genetic variants across about 100,000 that we looked at that were associated with whether or not an animal stayed healthy or wasted, at least in this sample size, which could be too small to detect that, but still an important result. We also had a nice opportunity to sample sea stars that were in the wild, Pycnopodia helianthodes, the sunflower star in Alaska, just as the disease was reaching that area. So we had naive populations as well as sites where there was wasting individuals and ones that were healthy. So we had exposed, so those were the ones that were still healthy, and wasting individuals. So we looked at these naive, exposed, and wasting, and again looked at the microbial communities in them. You can see that the naive, there's very stable, very simple microbial community composition in these sunflower stars. But as soon as they are exposed, even though they are showing no signs of wasting, the community starts to turn over. In particular, there's this dramatic increase in Vibrio, a particular kind of microbe. And then by the time that they're wasting, it's just a crazy hot mess, if you will. There's many different taxa that show up and are perhaps a result of this microbial or are a result of this microbial dysbiosis. So if we look at this particular Vibrio taxa, we can see here's this particular Vibrio group in the naive individuals. Here's their distribution of abundance. And again, this is on a log scale, so it's a wide range. But as soon as they're in this exposed category, there's a dramatic increase, over 1,200-fold increase. And that stays high once they're wasting. So this could be kind of like a smoking gun, something that's worth following up on. And here is one more of these kinds of principal component type analysis that I wanted to show you that has each one of these dots is from a different sunflower star. You can see that the naive individuals are much more clustered closer together and closer to the exposed individuals. And then there's this turnover that occurs from naive to exposed to wasting as the community changes. And these become more spread out from each other, the wasting individuals, because they're so different. There's just so much diversity happening. So to summarize, it looks like hypoxia is happening, Vibrio may play an important role, and that the microbiome may play a role in both being healthy and succumbing to wasting. So to summarize all of these things that I've told you, take-home messages are that large populations and living in a variable environment are important for being able to have adaptive genetic variation that can allow you to survive future global change conditions. And that there is capacity for and consequences of rapid global change adaptation. And lastly, that microbial imbalance, hypoxia, and perhaps Vibrio may play important roles in sea star wasting disease. But I can tell you that when I watch a movie, or if I go to a great talk, or if I go to stand-up comedy, I can't tell you what the movie was about or what the talk was about unless I took notes, or I definitely can't repeat a joke. So what I want you to leave with here, if you've gotten nothing else, are two things, both hope and resolve. So hope, because I show that there are mechanisms, there is variation in the genome, in the epigenome, in the microbiome for being able to survive these environmental stressors, these future global change conditions. But there's also potential costs. So we need to be able to do everything that we can do to turn back or at least halt global change conditions, to be able to allow organisms in this incredible biodiversity that we have on this planet to continue. But perhaps more importantly, I hope what you've seen through this talk is that it's the people, it's the process. So doing science is a community. So it is a community like we are here all today. And it's all about the people, actually, is what you need to take home. And it's all about doing what you need to do to maintain your spark. So thank you so much for your attention.
Right, that's that. And those takes ice cream to talk about.
What's that?
Takes ice cream.
It does, it does. You can
Yeah.
see we like ice cream parties. And this is a Jell-O, yeah, something Jell-O, and it was amazing. Yeah, happy to
Thank you,
take
Melissa.
any
That's wonderful. We have some time for discussion, questions, comments. That's why we're having the meeting, so we can all participate. I'm holding this box because it's actually a microphone, and then you can be heard by people who are not in the room. I'm going to pass you the microphone. If you can hold it close to your mouth so that it's picked up by the Zoom. I can actually throw it at you. Ready?
Wow. I had no idea it was going to be this fun. Okay, so for the layperson, what is interrelation between temperature increase, ocean temperature increase, and pH? Or are they separate forces? Are they interrelate?
Yeah, that's great. So it's a question of scale, right? So globally, they're definitely related, but locally, you can have variation in temperature and pH actually somewhat independent. globally, we have temperatures increasing, but we get a lot of pH variability, at least in the California current ecosystem, through a process called upwelling that actually brings really cold, nutrient-rich, low pH conditions. So in that sense, for a given environment, you're not having a positive one-to-one relationship there between temperature and pH. But on a global scale, they're absolutely related. Yeah. Yeah. Oh, yeah. So, and then I guess when we're thinking about the copepods, those can, all depends on the ecosystem. So I was thinking of the urchins, that those are here. The copepods, we've mostly studied on the US East Coast. And their temperature and pH can be more correlated, can occur. But these copepods are kind of near shore estuarine, and they experience pH variability all the time, up and down quite dramatically. So as you saw in those results, there was kind of no effect of the pH on them. But the temperature, it's quite a strong latitudinal gradient on the US East Coast for temperature. So that was the much stronger selective force. So yeah, it depends on where you are, what the organism experiences, because it all comes down to that level that they are experiencing it. What's their concert hall like, if you will? Yeah. Mm-hmm.
I'll ask a question so long. So the microbiome, it's pretty cool. We're learning about it in many ways. And you showed a distinct change in microbiome with disease. Do you have any idea whether that was the cause, or was that the result of the disease?
Yeah, absolutely. Great question. Chicken or the egg, cause effect, it's hard to tell. I think that it could be both. I think that it actually can be both. A pet hypothesis might be that there's low oxygen waters in the environment that get flushed up and could result, or say there's a phytoplankton bloom that all dies, microbes eat them up, makes that water pocket hypoxic, that washes over them, changes the environmental conditions around the sea stars, that then changes their microbiome because it allows for these anaerobic, microbes that love low oxygen environments to proliferate, and then that results in this turnover in the microbial community that results in, as they're trying to deal with this by maintaining their tissue integrity and fighting off with immune response, a snowball effect happens and they end up wasting, and then those microbes slough off and affect other organisms. So it's a really unique way to think about disease, but it's something that we should probably be thinking about much more as we think about wildlife diseases. Because it also happens in humans, but the things that get attention are the things that are easy. Like, oh, it was coronavirus, or oh, it's, so it's easy. When it's easy to like pin something on it. When it's complex, like perhaps sea star wasting, it's a little less satisfying, but it's still something that we need to dig in and figure out what's causing it. So I think we should put hypoxia or oxygen meters out everywhere and figure this out.
We had one brave layperson asking a question. So you're all welcome to ask questions. Curiosity is what we're looking for. Be quiet. Take care.
Always willing to ask, right?
Yeah.
Okay, so big picture question about temperature. Over and over and over, hearing how important the temperature is, right?
Mm-hmm.
It compares to pH or compares to oxygen, temperature.
Mm-hmm.
So what is it, right? Like, or do you have thoughts about that? Is it, temperature so important for like really incredible, functioning, or like, I don't know, you know what I mean? Just in a broad sense, do you have any sense about like why the temperature matters so much?
Yeah, why temperature and not other things?
Yeah.
Yeah, so that's a great question. And that's, temperature affects everything at the cellular level. So just to be able to, to run your metabolism. It's going to affect your metabolic rates will increase, which means then you need to eat more, which means then you're devoting more energy and time to eating and consuming and metabolizing that food, which means less time for reproductive efforts and things like that. So temperature is just a very fundamental stressor. pH can be a little bit different because it depends on how the organism regulates their pH. So it's not necessarily as universal as temperature. The same can be said for salinity. Some organisms regulate their salinity quite differently. So the temperature is, a very important factor, but I can say that in different organisms, it might be pH. So with the sea urchins, we focused on pH because a number of studies have found that that's quite important for being able to survive. That can cause more severe impacts than temperature. So it also depends on the organism, but if you're going to make a blanket rule about about animals, at least I can say temperature would be more important just because of those metabolic costs. Yeah. Hey Sam.
Hi, great talk again.
Thank you.
I was wondering about some of the data you showed for the copepod selection experiment. I think you had like bean fitness as the response, and so I'm wondering, as an individual response, I'm wondering if the recovery that you saw in fitness led to population-wide recovery, or whether you were able to measure that at all.
Oh yeah. Yeah, that's great. I don't know if I put the slide in here. So we did look at that. Well, oh no, I don't have it. So we see a dramatic decline in the population fitness when we transplant them from ocean warming and acidification back. So if I could draw, it would look like this. So the fitness for ambient animals is here. The fitness for ocean warming and acidification is a bit lower. And then when you transplant the ambient animals into ocean warming acidification, it stays the same. You're fine. When you transplant these lower fitness ocean warming and acidification, their overall fitness is still high enough to sustain the population. But it is lower. But it declines when you transplant them dramatically. And that decline, we did another experiment where we raised them in low food environments. That decline becomes much steeper and lower when they're food limited, as opposed to when they are food abundant or replete. Yeah. Does that answer your question?
Yeah, thank you.
Yeah, you're welcome.
Fantastic.
Hi. Do they use sirtuins like humans do to regulate what parts red?
I'm sorry.
Do sea stars and urchins use sirtuins, the same kind of mechanism to unspool the DNA and decide what parts are red, or is it a different mechanism?
Like unspool from like histones and unpack the chromosomes.
Yeah. Like
and
which
all that.
genes are red? This is a cell that's like for chitinous, whatever.
Yeah. For sea urchins and sea stars, I believe they do. They're in the deuterostomes. So I think all of that sort of like basal functioning is pretty much the same. Yeah. So I think also for epigenetics, it might be similar mechanisms. Yeah, Vicki.
I am not a scientist. I'm an artist and a graphic artist and a writer. So I ran into somebody this afternoon in the lab, and I was saying that some of the research here is on the purple urchin. And their question to me was, why is that important? So I guess the question is, from a lay perspective, if the sea urchins are in trouble and they start going away, how does that affect the rest of the environment?
Yeah. Well, now that the otters are recovering, it is prime food for the otters. So we definitely need and want them. But as we're in this kind of limbo where we have removed a lot of their predators, spiny lobster and large fish, And with wasting disease, all the sea stars going away, there's definitely an imbalance in the ecosystem. So now they're kind of the nemesis, as I was mentioning. So what would happen? Yeah, there would be a problem. There would be an ecological cascade to them being gone. So, for example, Anya and I were talking today about corals and algae, and in other ecosystems, urchins are really important for grazing and keeping algal growth down at bay. So they're going to be critical for coral reef health and function in the future. Our purple urchins, they might be critical for other microorganisms or other making habitat available for other organisms in a similar way that sea stars do, or these other urchins in tropical ecosystems. And the reason that, so I guess I'm not studying them for a conservation reason in the same way that, studying sea stars or even studying the copepods as basal to the food web. The reason I mentioned the power of studying them is due to their natural history, the fact that they have this broad geographic distribution, the fact that they have this high dispersal potential makes them a great model for understanding how they might adapt. And that can then be translated to other organisms. And we can say like, oh, large population sizes are important, or if you live in a variable environment, you're going to be better off. And we can kind of take lessons learned from purple urchins and apply them to other organisms. That's kind of, thanks for asking that question. That points out the importance of doing basic science research that doesn't necessarily have a direct translation to conservation or, etc. Thanks, Vicky.
That's the left-hand side.
Hi there.
Heads up.
My question was, so I know at some point during your talk you had mentioned that you guys were looking to answer the question of how sea star waste disease went from something that was kind of just seen sporadically to such an epidemic that it is now. And maybe I missed this, but was there a consistent to that question?
Yeah. Good question. So I think once we know more of the actual cause, then we'll get to that. But I would say that if we could, based on we're marching closer, and if we could figure out if it is hypoxia by putting out oxygen sensors in habitats where sea stars live and slightly further down, and get at that, because temperature is easy to measure. So we're always trying to see, is it correlated with temperature? In some sites it is, some sites it's not. So it's not temperature. So oxygen, seems to be, at least based on our results and the results from other groups, seems to be what could be causing it. So maybe there are things like, by say, I mentioned agricultural runoff. So let's just say there's more runoff happening into the oceans that results in phytoplankton. It's nutrients for phytoplankton. So then there's a phytoplankton bloom that then after they've consumed all those resources, they die and they sink. And then that's energy for microbes. And those microbes eat all those dead phytoplankton. And then that results in a really low oxygen environment. And then that gets washed up and causes this cascade. So it could be that maybe that's the cause, but we're not doing enough. I mean, this is a great question to plea for more environmental monitoring, to be able to try to figure out what are the potential causes happening. And maybe we have, to your question of why more now on this tipping point, or you pointing out my question, is that maybe we have more of those kinds of things happening, more of these phytoplankton blooms happening now than we ever did before, and it's more localized and regional than it ever used to be, which, with the intensification of agriculture and things like that, this is not beyond the realm of possibility.
Yet, what's the most current data on percentage of loss of sea star life and also bull kelp life, especially here in Northern California?
Sea star life and what?
And bull kelp for it?
World Cup. Ooh. You know, Eric would be the better person for that. But I think, and Jackie, there's recovery is happening. So we're seeing a lot more juvenile sea stars come up from either wherever they were hiding or, you know, further deep in the ocean or from cracks and crevices, and having space to then grow and not being outcompeted. And in one study, we looked at the microbiome of juvenile versus adult sea stars, and we see that they're quite different. So there could be some difference in resilience, and also related to the microbial community of these juvenile versus adult. So there's definitely recovery. And I was talking with Jackie recently too, and she was saying that we also are seeing, you know, this way that that the sea stars die is a way that they're amazing at regeneration. Sea stars are. That's one of the reasons that they're studied. And I love this example of bald eagles in Alaska use sea stars as kind of a renewable food resource. They'll go chomp off an arm. That's a tasty bite. The sea star doesn't die. It grows back an arm. They can just do that. So this and this is also the way an old sea star dies. When it gets old or something else happens, they can get these lesions and they can pull themselves apart. So all that to say that Jackie was saying that we're seeing more recovered sea stars that had lost an arm, perhaps due to wasting, and have grown it back. So either there could have been some immune priming, so maybe they were one of those ones that activated an immune response, and they are able to better respond the next time they're experiencing a low oxygen environment or whatever the pathogen might be, and they're surviving better and they're regrowing. So bull kelp, I don't know. I can't speak to that, but maybe they're being impacted by the overgrowth of sea urchins, and I don't know that is. I know there's been lots of smashing of sea urchins. If you want to smash the sea urchin, just send them to me instead. I'll take them. We have a refuge in Vermont. Yeah. Oh, the online.
Yeah, we have one question from the online chat here from Debbie Hunter, and this is the question that's asking about some of the complexity of climate change that kind of ripples through the food web. So it seems like temperature will really impact the phytoplankton community in terms of diversity and abundance. This would impact the copepods if they could adapt to this changing food source. Would this impact the copepod's overall fitness, yielding a genetic response? Would this express as exaggerated change in the copepods because temperature affects not only the copepod's metabolism, but also.
Yeah, great question. And that's actually, we don't have any experimental data on that yet, but that's one area that we're looking at, is how these trophic interactions can be impacted. So if when you grow phytoplankton up in warmer temperatures or even higher or lower pH, they're happier. So, or as the abundance changes, so that can definitely affect their fitness, and could in turn affect the fitness of the organisms that eat them. It's just that, you know, these ecosystem effects are so interconnected and get hard to predict. So doing bits and pieces of lab studies and then also measuring patterns in nature is the way to go. But we're kind of, you're thinking way ahead of us. You're there before we are. So that's where we're going next. There was one more question. I don't know if we want to, one more community question.
I'm going to be quick.
So I was wondering about the preferential recovery, where it seems like Physaster are coming back, but the Pycnopodia, which we really need to control the purple sea urchins, are not at all. So is there any information about why that might be?
Yeah. So one hypothesis is that the susceptibility to whatever it is that causes wasting could be related to their what's called rugosity, so how bumpy and wavy their skin is. So the more kind of soft and bumpy and wavy their skin is, the more susceptible they are. So Pycnopodia would be on that way end extreme of fluffy, bumpy, soft, high rugosity organisms. Pysester would be a little bit under that, all the way down to the bat star, et cetera, and those are least susceptible. So there is a relationship between that rugosity and susceptibility, and that also fits with the microbiome and hypoxia hypotheses, because a lot of those microbial changes are happening, like what we sampled, the tissue we sampled was just a little tiny bit of that skin sample. So that rugosity keeps allows microbes to proliferate right there, and then also lowers the oxygen levels in this really tiny microenvironment just along their skin, which then can lead to this, cascade of microbial turnover and hypoxia that is just acutely experienced by any, individual sea star. So yeah, figuring out, what's causing it is going to help all of the organisms, but those with higher rugosity, bumpier, soft skin even more. Yeah, great question. Yeah.
Thanks, Melissa. I'd like to invite everybody to join us for a light reception and continue some of these discussions if you like. We'll talk to each other. Please, everybody, feel welcome to stay for a little while. And then finally, Melissa, thank you very much for a wonderful talk.
Yeah, thank you. Thank you.